an enzyme has the suffix "ase." And so like "-ose" was for sugars, "-hol" was for alcohols, things like that. Enzymes end in "ase." This is how you know that it is an enzyme. So peptidases, amylase, things like that. Alcohol dehydrogenase. If it has ASE, ase, it is a catalyst. It is an enzyme. Sorry, it's a protein catalyst. The way the names generally work, however, is wonderfully convenient. It tends to have two parts. The first part of the name is the substrate, and the second part of the name is the reaction. So names like oxidoreductase. Well, oxido is going to be oxidation, and then reduction, reductase. This is an oxidation reduction enzyme. This moves electrons. You can also have alcohol dehydrogenase. It dehydrogenates. What will happen for this, if you have ethanol, this will react to give you, well, eventually, acetic acid. There's a few steps in there, but the idea is it breaks down ethanol and turns it into another molecule your body can use, in this case vinegar. We use lots of acetic acid for various things in our body, and we can also urinate it out. It's part of why people get hangovers. If there's a buildup of acid, it's not great. It interferes a bit with your brain and makes people feel ill. There's plenty of types of enzymes. You can have transferases. Transferases move functional groups. So methyl transferases are pretty common for moving a methyl group, a CH3, around. We tend to rip them off one molecule, put them on another. We use it on DNA all the time. It's part of how we turn on and off genes in our DNA. Hydrolases. A hydrolase can add or remove water. You can have things like lyases. Your lyases tend to add and remove double bonds. So these are good for fat saturations. You can have isomerases. Isomerases change between isomers. These are very good for converting sugars. Remember, the only difference between a galactose and a glucose is which direction those OHs are going. This can convert D to L or L to D, depending on what isomerase you're using. These also affect things like amino acids, to make sure they're facing the right direction. So these affect chirality. Now, there's many, many more, just to give some examples of the type of things that these can do. Basically, any possible reaction can be mediated, and probably is, by some enzyme in the body, made to go faster this way. Sometimes, you have a protein, got some alpha helixes, beta sheet, alpha helix. Every once in a while, that's not enough. You need more than the protein. Sometimes there is just some other molecule. Hemoglobin has a heme with iron. Heme is this ring structure of carbons with some nitrogens that attach to the iron. You can have biotin, you can have NADH, all sorts of various things. So there are extra pieces. Those extras that are sometimes needed. A metal ion, a sulfur iron cluster, a heme. There's a lot of different extras. If they are organic, they tend to be called coenzyme. And if they are inorganic, they tend to be called cofactors. Adding separation, as we've learned, there are more and more things that attach to the protein or work with it. And so sometimes you just need more than the protein. often the protein for the enzyme is just there to hold the other pieces. And then you stick to the metal. In fact, about a third of all enzymes have a metal ion that is where all the chemistry happens at. Ends up if you only have nitrogen and oxygen and hydrogens, like surely there was 20 amino acids, but that's still not a lot of variety. Adding a metal means now you have many metals that can be attached to all those oxygens and nitrogens, and so you get a more varied possible reactions. So there's a lot of cofactors that get included in enzymes at times. Critical ones that come up a lot: iron, so Fe2+ or Fe3+, depending on what they're doing. So you need that in, like, hemoglobin uses iron. You've also got some calciums in photosynthesis. You've got some manganese or zinc. Superoxide dismutase. In humans, one of them uses these manganese and zinc ions. So what is superoxide dismutase? Well, it breaks up superoxide. Superoxide, O2 with a single minus. This is oxygen. You have minus here. And it doesn't have all of its electrons. There's an unpaired electron. This is a free radical. If you ever see health info talking about antioxidants helping you protect against free radicals, this is usually what they mean. Because we breathe dioxygen, we just occasionally on accident add an electron to it. And so as a result we make superoxide, which is super destructive. It is unbelievably destructive to our cells. So we have an enzyme whose only job is to break it up, make it go away, put an extra electron on it or rip one off, whatever it takes. And that uses these manganese and zincs. The organic atoms themselves don't have spare electrons to do that with. Instead, what can happen is manganese 2+ will react with it. So O2 with minus, you get manganese 3+ and O2 2-. Now it has the extra electron. Now it's not a free radical. The next time one comes by, it runs into that 3+, reacts with it, and the manganese steals the electron, which releases just regular oxygen. And so we're used to regular oxygen. O2 2-, this is peroxide. And our body's actually used to that. That's more stable. We can tolerate it. We have other enzymes that deal with it. It's not very destructive by comparison. And so this is a back and forth. One time it gives away an electron, one time it picks it up, but it's really the metal doing it. The enzyme is just a protein holding that metal in the right position. And so there's plenty of times that we mix other components in rather than just the protein.